Here we are going to see the chapter Heredity. We have to know what is heredity. Heredity is the passing of characteristics from one generation to the next generation. This process is done by genes. Okay, it defines the characters in the organism. So it is done by two ways. One is the asexual reproduction and second is the sexual reproduction. So we are going to see the differences between the both. So first, the young ones resemble each other with a single parent. Here it has only little differences and only one parent is involved here and no gametes are involved. But in sexual reproduction, young ones are not identical. Here the differences are there but it is visible among themselves and their parents. Here the two parents are involved and the young ones also receive both the characters from the father and the mother. So male and female gametes are involved here. So these are the differences between the asexual reproduction and sexual reproduction. So the variation is nothing but it is the differences shown by the individuals, also by the siblings of the same parents. This is the variations. This is occurred during DNA copying. This DNA copying is nothing but it is the process of producing two identical copies from one original DNA molecule. Okay, there are two identical copies. This biological process occurs in every living organisms. So we'll move on to the next. Here, accumulation of variations in asexual reproduction. Okay, here as discussed earlier, only one single parent is involved here, and the single individual reproduces asexually two individuals. Okay, so this is a single parent, it reproduces two individuals. So again, next these two reproduces other two to form four individuals. All these individuals will be similar, but minor variation, that is the difference, occur due to inaccuracies in DNA copying. Okay, next we are going to see Mendel's work. Georg Johann Mendel, known as the father of genetics, he was an Austrian monk. He worked on pea plants, okay, to understand the concept of heredity. So this pea plants, the scientific name of this pea plants is Pisum sativum. He he laid the foundation to the modern genetics. So he had three basic laws for this inheritance. He used these pea plants to find all these. So first law of dominance, second one the law of segregation, and third is the law of independent assortment. So next we're going to see the Mendel's law of inheritance. Here in the first law of segregation, only one of the two gene copies present in an organism is distributed to each gamete that it makes. And here the allocation of the gene copies is random. So for example, in a pea plant, let's consider two alleles present. So the capital T represent tall and the small t represents short. Here two alleles are present. During segregation, they segregate during the formation of gametes, okay, as capital T and small t. Here only one will be carried out. And in the law of dominance, here when the parents with contrasting traits are crossed together, only one form of the trait appears in the next generation. So here in our example, the capital T will be the dominant factor and the small t will be the recessive. Okay, so law of independent assortment. Here in this law, when two pairs of traits are combined in a hybrid segregation, here the one pair of characters is independent of the other pair of the character. They are independent, they are not dependent. Okay, the father of genetics, Mendel, he selected the garden pea plant for his experiment. Here the scientific name of this garden pea plant is known as Pisum sativum. Here he used a number of contrasting characters for the garden pea plants. Here there is a table. Okay, here in this table there is a list of contrasting characters for the garden pea. So this character may be a flower color, position, or seed shape, or it may be a height of a plant. So he used 7 pairs. So here the dominant traits and recessive traits are listed, so you can have a glance. So next we can come to know what is dominant traits. They express themselves in an organism and they can be seen visibly. For example, it may be a tall pea plants. Okay, so in recessive traits they are present but not expressed in the presence of dominant allele. So for example, already we have discussed. So let's consider 3 possibilities for this garden pea plants. Here 2 capital T. As discussed, capital T stands for tall. So here the plant is possible to be tall. So next combination is capital T, small t. Here this T, capital T is available, so it is tall. So the next possibility is small t. So here both small t is short and it is recessive. Let's consider the Mendel's experiment with the example. So in the parent generation, let's consider the pure dominant yellow pea plant is cross-pollinated with the pure recessive green pea plant. Okay, here when they are cross-pollinated, we get 4 hybrid varieties, that is the same type in the F1 generation. Okay, they are again self-pollinated, and in the second generation we get 4 varieties. Okay, they are in the ratio 3:1, that is 3 yellow pea plants and 1 green pea plant in the F2 generation. Monohybrid cross. Here it is the cross between the individuals of the same species in which inheritance of the contrasting pair of single character is considered. As the name indicates, mono. Mono means single. Single character is considered here. Next one is the dihybrid cross. As the name indicates, di. Di means double or 2 characters are considered here. It is a cross between the individuals of the same species in which the inheritance of the contrasting pair of 2 characters are considered. So let's consider an example for monohybrid cross, that is 1 character. They have taken here as the height of the pea plant. And in dihybrid cross, that the character considered here are seed color and the seed shape. Okay, so here let's see the ratio. The ratio obtained in the F2 generation when a monohybrid cross made is 3:1 in a phenotypic ratio and 1:2:1 in a genotypic ratio. Next, the ratio obtained in F2 generation when a dihybrid cross is studied is usually 9:3:3:1 phenotypic ratio. So please remember this. As already discussed, these are the conclusions of the Mendel's experiment. So the first point is capital T. 2 capital T's and capital T, small t, both are tall plants when capital T is available. So next, while small t, 2 double T is a short plant. Okay. So second point is a single copy of capital T is enough to make the plant tall. So here the capital T is the dominant one, while both copies have to be small t for the plant to be short. Okay, next one is the third. Character or traits like capital T are called dominant traits because it expresses itself as discussed earlier, and small t are recessive trait because it remains suppressed and it is not expressed properly as such of dominant trait. And from these observations, Mendel put forward the rules of inheritance. Now we have discussed the examples of the pea plants. Now we are going to see about the human beings. Here the 6 chromosomes. In human beings there are 23 pairs of chromosomes in our body. Out of these 23, 22 chromosomes pairs are called autosomes. Okay, and the last single pair of chromosomes, it helps in deciding the gender of that individual. So this is called the sex chromosome. Next is the sex determination. The mechanism by which the sex of an individual is determined is called the sex determination. So the different species use different methods. So some rely on the environmental factors. So when we consider the example of crocodile, okay, here the sex of the young ones will be determined by the temperature at which the eggs are kept. So let's see, when the temperature is cool, around 30 degrees Celsius, the hatchlings will be female. So at warmer temperatures around 34 degrees Celsius, they hatch all males. Okay. So in some organisms, individuals can change sex, which shows that sex is not genetically determined. So example, let's consider snail. So when the availability is all female snails, there is a chance they can change by themselves to male. Okay, this is the example. So next, the sex determination in human beings. So human female have the X chromosome in all the gametes produced. So whereas human male have two types of gametes, okay? One is the X chromosome and one type of gamete with Y chromosome. So let's see in the next slide in detail. So next we are going to see the sex determination in human beings. Here the parents, that is the father has the XY chromosome and the mother has the XX chromosome. Here, if the male gamete carrying X chromosome fuses with an egg, the young one produced will be a female. So if the male gamete carrying Y chromosome fuses with an egg, then the young one produced will be a male. So zygote form after fusion of gametes. Okay. So there is a 50% probability of a female child in this case, and here the possibility is 50% probability of a male child. So these are the important definitions that are used in this chapter. So you all have a glance in these definitions. Thank you.